so easily. I don't know what has happened. Anyway, no problem. We'll sort out this. Let's go back to our presentation. Topic 2 is on recombinant DNA. And let's go back actually to when we were doing the history and evolution of biotechnology. SMR402. I'm going to sort out these problems. So maybe in the next lecture, we are going that we did on to do this. Okay, so genetically modified organisms. If I touched a bit on recombinant biotechnology started really over 6,000 years a little bit more and deeper into that. And it started as a very simple thing, just a fermentation process. Maybe this time round is going to be even more clearer to you. So it's not necessarily a new thing that that I'm introducing. I hope my screen is big enough. Can you all see it? Can you see my screen?
Yes. Can see.
Yes.
Yes.
Okay, sawa. So what is recombinant DNA? As you can see from the screen, recombinant DNA refers to the creation of new combinations of DNA segments that are not together. When in SMR 301, there was this topic on genetic improvement of fish. And we went through the product improvement mechanisms or strategies. We talked about crossbreeding, we talked about hybridization, we went for monosex populations. We also now looked at polyploidy, we looked at chromosome manipulation. But I remember telling you that all along, in all those things that we did not touch the genes on the genes. Okay, so you need to be very, very careful and know that as we are actually going to look at it later, that there's a difference. In recombinant DNA, you're basically taking DNA from one organism. It could be a plant, it could be an algae, the isolation and manipulation of genes allows for more precise genetic I want you to appreciate is that this technology is so broad. There is so much you can do with it. And it's now touching on everything, to agriculture, to industry, name it, it's everywhere. Gene of one organism being found in another organism. So when you talk about recombinant DNA, what are the basics? What are the basics of recombinant DNA? The basics are you identify a gene of interest. For example, maybe it's a gene for helping a plant not to freeze during winter maybe a certain plant has that gene. And maybe you'd like to put that same gene in your fish that you're culturing over winter so that they don't freeze. So you identify your gene of interest. Then you isolate it because you don't need the whole, remember, so when you go to the gene, you go through the cell, you go to the double strand helix, then you identify your gene of interest. And once you say this is the gene, maybe it's CTTR, that's your gene of interest. That is the gene that is responsible for this plant not freezing or for this fish not freezing. So you isolate it and then you cut it with restriction enzymes. Restriction enzymes are called the scissors in this work. And I think we'll see a video later on so that you can visualize how this happens. So you cut this with restriction enzymes. And then once you do that, you look for a vector or a carrier, which in most cases is a plasmid. We are going to look at all that, don't worry. And you put it, you ligate, or to ligate is to join, to put together, okay? You ligate it into a cloning vector, and then from there, transform the recombinant DNA molecule into a host cell. So it carries, a host is the one that carries. And then from there, once you are sure that the new gene is already combined with the host cell, from now you can think of multiplying it so that you can have many genes of interest. Remember, when after you clone, you want to produce as many as possible. So what you do, you put it, mostly they are put in bacteria, and then they are produced in millions. So you can say, okay, now we have a gene for antifreeze. We have produced it, it's here, we can use it. But this will become clearer. Let's see whether this. So let's look at this. This is a human cell. So what you're doing, you are looking for the gene for the human growth hormone. And this is the gene. From this cell, you go to the double, strand, the chromosome strand. You look for this particular hormone. You have already identified it because maybe you've already done your gene sequencing. You know, this is the one that is responsible for the growth hormone. And then from there, you cut. If you can see this little bit of strand has already been cut, you cut it using enzyme. So this is, when you cut it, what you normally have, you have what we call sticky ends. So there's this now you need to put it somewhere in a vector. So this is the time you look for a bacterial cell. In the bacterial chromosome, in most bacteria, you get what we call it's a plasmid. They are very good carriers of genes. So what you will do for the plasmid, because you need to fit a new gene here, you're going to cut this depending on the space of this. So you're going to cut this, so you'll end up with this. This is the plasmid minus that part that has been cut out. And what you just want to do is to create room for this DNA for the human growth hormone. So once you have a plasmid which is ready, this plasmid over here I'm pointing, this one has been prepared to receive the human growth hormone. And the human growth hormone has already been cut or excised from the human DNA, and it's ready to be joined. So you take this piece and you put it here in the missing link. Can you look at the sticky ends? This is a sticky end, the ones which are yellow. This is also a sticky end. Just a minute. If you look at the plasmid, also it has sticky ends. These sticky ends are very important because that is where the new gene is going to fit. And as you can see, once it is put, it fits perfectly. The sticky ends join together. And that's the process of ligation. Of course, at times they need to put something else. You need to put something else. You'll find out later to make it even stick firm. Then from there, you take your plasmid plus the inserted human DNA, and you put them again in a bacterial cell now. And what you want to do is to replicate. Now you can grow these bacteria in agar. Remember when we talk about agar that we get, mostly we get from seaweed. Now this bacteria, you see, you can return, or you can get, and you can put the plasmid with the human growth hormone back into a bacteria. And then now you can culture these particular bacteria using agar. Am I clear? Is there anything that someone doesn't understand up to that point? Because it's very important that you understand. It's not difficult, but you need to understand. Anybody who is lost? Is there anybody who has no idea what we are doing? No? Omoenga Derek?
Yes, Madam Amin.
Are you okay?
I'm okay. I'm following.
Okay, great. So, now let's get to the proper introduction. So basically now this is the write-up of what I was saying. That once you identify your gene of interest, you know where you want to get it from, if it's a human DNA, if it's a plant DNA, if it's an animal DNA, you know where to get it. You've already identified it, then you need to extract it. Now, they are different. Remember, when you want to extract the DNA, you have to break through into the cell, because that is where the chromosomes are, that is where the DNA strands are. Of course, on the DNA strands, that is where the genes are. So you have to break through your wall, the cell wall. So most nucleic acid extraction techniques involve steps to break open the cell, and then to use enzymatic reactions to destroy all the desired macromolecules. Remember, a cell has so many things. It has mRNA, it has DNA, it has the cell wall, it has the Golgi bodies, all those things that are in the cell. But remember, what you're interested in is the gene. So you're going for the double helix. Okay, going for the double helix. If you're going for that, then you don't need all the other stuff that is in the cell. And that is why you have to destroy it first, so that you can access the genes or the chromosomes, so that you can access the genes on the chromosomes. So every other macromolecules has to go, and you can do this using enzymes. At times, cells are broken open using a detergent solution containing buffering compounds. So, let me reduce this so that you can see. So look at DNA extraction. So you've got your test tube there, you've got whatever cell that you want. So once you put them in the first test tube, you can use a detergent to lyase. To lyase is to is to break open. So you break open those cells using a detergent, because what happens with the detergent, it disrupts the plasma membrane. And I think even for COVID, One of the reasons you just keep washing your hands with soap. That is what the soap does. So you open, you lyse open the cells using a detergent. And once that is done, all the clear contents that remain are treated with protease to destroy the protein and RNAs and to destroy RNA. Remember, this is where now you put the enzymes, because you want, these are the macromolecules in the cell. So you have destroyed the cell wall. Now you want to destroy the other stuff, the other cell contact. So you can use the RNA, which is an enzyme, RNAs, to destroy the pink part. Then you've got this creamish part, which is the debris. So cell debris is pelleted in a centrifuge. I hope you all know what a centrifuge is. A centrifuge is that machine that really shakes and separates. It tends to separate the contents, be it of water, of liquids, or whatever it is. So the supernatant, which is the liquid containing the DNA, is transferred to a clean tube. Remember, the DNA now will be contained in the liquid. Then whatever other stuff, probably the solid, is cleared out. You don't need it. So once you transfer the DNA into a clean test tube, the DNA is precipitated with ethanol. Remember, it is liquid here, but once it gets here, you can precipitate it. Make it. You know what precipitation is? Making it sort of solid. It's precipitated with ethanol, and it forms a viscous strand that can be spooled on. You see, like, to spool is to tie it round. So this is something which is viscous. It's it may not be necessarily liquid or solid, but it's there. It's there. You can, as you can see in this case, actually with the DNA, you can already spool. You can tie it around a rod. So let's go back. How do you cut DNA? So DNA can be cut into large fragments by mechanical shearing. To shear is to cut. Restriction enzymes are the scissors, as I had said before, of molecular genetics. So in molecular genetics, once you want to cut something, go for your restriction enzymes. Those are your scissors. Restriction enzymes, or further on referred to as RE, are endonucleases that will recognize specific nucleotide sequence in a DNA and break the DNA chains at those points. So remember, restriction enzyme is very specific. Once you send it, once you put it, wherever your DNA is, remember this is a chromosome. There are so many DNA sequences, but the restriction enzyme is coded in such a way it will recognize your DNA of interest. And once it recognizes it, that is what it will cut, nothing else. It's going to leave everything else intact, but it's going to go after your DNA of interest, and it's going to break it at the right point. So we are being told that there's a variety of restriction enzymes that have been isolated and are commercially available. So I think over time they've developed these enzymes. They are there if you want to cut this enzyme in a, maybe it's the growth hormone, the antifreeze gene, then there are specific enzymes. Some of them you're being told they're already commercial. Maybe you can just go to the shop and say, I want this restriction enzyme that I can use to cut this particular gene. So what is cloning? Cloning allows for the creation of multiple copies of genes, expression of genes, and study of specific genes. Cloning basically just making copies. Making copies. Cloning is simply making copies. It could be copies of genes. It could be copies of expression of genes, and also help you. It will also help you in the study of specific genes. Remember, when you're studying genes, you can't use just one gene. You cannot only use one gene when you are studying genes. You need very many genes. So will you keep going to look for restriction enzymes, cutting the gene, and that kind of thing? No. Once you cut your gene, you isolate your gene of interest. The next thing you want to do is to make as many copies as possible so that it will help you in whatever work it is. Maybe if you want just to study, if you're just interested in carrying out research, that will help you. If you want maybe to put it somewhere and do experiments, then you already have your DNA. So to get the DNA fragment into a bacterial cell in a form that will be copied or expressed, the fragment must first be inserted into a plasmid. That one we have said. So that is the second step. The first step, I keep repeating, identify your gene of interest, isolate it, cut it using restriction enzymes. From there, insert it into a plasmid. And I told you plasmids mostly appear in bacterial cells. So it's even expressed there. A plasmid or a vector, and I told you a vector is a carrier. A plasmid is a carrier. It's the one that carries for you your gene of interest. Okay? So it's a vector or a vehicle that carries the desired gene. And you've already seen it in the flow chart I showed you. It is circular. And by the way, a plasmid, even when it is in a bacteria, it replicates independently. So it doesn't need the chromosomal DNA in a bacteria. And that is why they are very, very important vectors or carriers. It's because once you remove it, once you put the gene of interest and put it in another bacteria, it will continue to replicate on its own. It doesn't need the DNA, the chromosomal DNA of a bacteria to replicate. Is that clear? I want to stop at that point and make sure we are all together. Anybody who is lost? Just put up your hand. Anybody who is lost, who doesn't know where we are, cannot make a difference between a plasmid and a vector? Come on, Puthia, go ahead. Come on, you raised your hand. You want to say something?
That point of independent of chromosomal DNA.
Please talk loudly.
That point of replication, independent replication.
Okay, yeah, I was saying why plasmids are preferred as vectors in recombinant DNA work and any other work that involves removing a certain gene from one organism and replicating it so that probably you can put it in another organism. Why a plasmid is chosen is, number one, plasmids will mostly appear, they're almost always in bacteria. And why plasmids are chosen is because they have their own independent replication. Remember, during cell division, remember during meiosis, mitosis, and the cell division and the replication of cells, it's always in the chromosomes. You remember how the chromosomes separate, then another daughter cell is formed, then it also separates and that kind of thing. For plasmid, it doesn't need the chromosomal DNA to replicate. It replicates on its own, which is very good because it will be able to do the work you want it to do for you. You can insert your own genes and still get what you want without touching the genes of the bacteria. Is that clear?
Yes.
Anybody else? Just raise your hand. I'll see it. Okay. Anybody else? Okay. So let's continue if everybody is happy. So once you've cut your gene of interest and you have identified your plasmid, what do? You want to join the DNA. In a typical DNA cloning procedure, the gene or other DNA fragment of interest is first inserted into a plasmid. That one, we know it already now. We can sing it. The insertion is done using enzymes that cut and paste DNA, and it produces a molecule of recombinant DNA, or DNA assembled out of fragments from multiple sources. I think we've also gone through that. Once you've got your gene of interest, you've got your plasmid, you have created space in your plasmid, then you can join these two, again using enzymes. Of course, the sticky ends are very important, but you also need enzymes to help you ligate or join. The word ligate means join. So I think this is very, very interesting, and I'm just hoping that some of you will end up in this field. So to join the DNAs, so this is how it happens practically. To join the DNAs, they are mixed together in a tube. Okay. So if both have been cut with the same restriction enzyme, the ends will match up. Remember, even for the plasmid, to make space for the new DNA, you also have to cut it. You have to cut it open. And I think in most cases, they advise use the same restriction enzyme for the DNA of interest and also for the so use the same restriction enzyme for the gene of interest and also for the plasmid. If you do so, then their sticky ends, the ones that I was showing, they will match. Okay. But the DNA ligase is the glue of molecular genetics. So there is also another enzyme called ligase. And this is what they use in molecular genetics to hold the DNAs together. DNA ligase creates phosphodigester bond between the two DNA ends. I think we already saw that, unless there's something, but we are also going to watch a video on this and it's going to become clear. So just be a little bit more patient. Let me see whether I can show the video at this point so that we don't. I'm trying to see where I put the video. I think I know where it is. Okay, we'll watch the video after. We'll watch the video. Let me finish the process first, and then we can watch the video, because I think I put it elsewhere. I put it in the. So next, the modified plasmids. The modified plasmids are the ones that already have your gene of interest. You have joined the two together. The modified plasmids are introduced into a bacterial host for replication. You put that again into a bacteria. This time the bacteria will be a host, and now you can grow the bacteria in nutrient agar. Bacteria carrying the plasmid are selected and grown up. So remember, once you do this, there are those who will be able to to produce the plasmid, others will not. So you have to select the ones that were able to replicate. This is what we are saying you select. And this is now what you continue growing. As they reproduce, they replicate the plasmid and pass it on to their offspring, making copies of the DNA it produces. There are people who are still joining now, so I still have to keep stopping to accept them in class. Okay, so DNA cloning happens when a section of the DNA takes place, when a section of the DNA that has been inserted into a plasmid, which is a vector, into a plasmid molecule, and then is replicated in a host cell to form many copies. It is a molecular biology technique that makes many identical copies of a piece of DNA, such as a gene. By the way, once you are able to make copies of this gene, you can even put them in a library. Yes, there's a DNA library. So that next time somebody wants the human growth hormone, they don't have to go through all this process again and say, oh, we have it. Here it is. So I don't know whether they are sold, but well, they are there. So what do you need in order to say this is a good cloning vector and this is not a good cloning vector? What do you need? What are the requirements? What are the characteristics? Just a minute. What are the characteristics of a vector? For example, a plasmid. Number one, it should be capable of replicating in host cell. Not all are able to do that. So for you to be able to get a good one, that's going to be important. Then it should be able to replicate in a host cell. It should also have convenient restriction enzyme sites for inserting DNA of interest. That means not all vectors have these sites. Okay, so for the one that you're going to choose, you must make sure that it has sites, convenient sites for inserting DNA of interest. It should also have a selectable marker to indicate which host cell received recombinant DNA molecule. Once you actually get this, once you get to insert the new gene, Not all plasmids. For example, now we're talking about a vector of plasmid. Probably not all plasmids we receive. Remember, this is a genetic way, something you're putting inside, you're shaking, and you're hoping that plasmids received the gene of interest. But not all plasmids actually receive. Some will receive and some will not. So for you to have a very good cloning vector or the right plasmid, you have to make sure that you can be able to, there has to be a marker in that plasmid that will help you know whether this one has an indication that it received the gene of interest or not. Okay, because if it did not receive the gene of interest, then you're not interested in it. Okay. Then the other characteristic of a vector or a carrier, and in our case, we're saying it's a plasmid, it should be small and easy to isolate. It should not be a complicated thing. It should be something that's totally easy to replicate. So let's look at bacterial plasmids. Where are they favored? Where are they good? Bacterial plasmids are small, circular DNA molecules that are separate from the rest of the chromosome. I think that one we have already established it. It has its own DNA molecules, but it's not part of the chromosome. They replicate independently of the bacterial chromosome. In a typical cloning experiment, a target gene is inserted into a circular piece of DNA called a plasmid. The plasmid is introduced into a bacteria via a process called transformation. And bacteria carrying the plasmid are selected using antibiotics. Bacteria with the correct plasmid are used to make more plasmid DNA, or in some cases, induced to express the gene and make protein. In some cases, you're not just interested in the gene. Remember, if you want to make a protein, so protein making is the next step after you get the gene. Maybe you want to produce a certain protein for whatever reason. So once you've got your DNA, once you've been able to replicate it, then you can go into the next step and make your protein. Okay, so make sure you've got the correct plasmid. Make sure the transformation process has taken place. And make sure you've been able to identify which plasmid carried the gene of interest. And in this case, they are saying you can select them using antibiotics. Probably we'll get into that, maybe not in this lesson. But you should be able to do that so that finally you can do whatever it is you want to do with the genes. What do you want to do with the genes? Do you want to make proteins? Then if you want to do that, then go ahead. In the next step, growing up of DNA. The inserted DNA fragment is copied along with the rest of the bacterial DNA. I think that is clear. And so I hope that is clear, because as the plasmid is replicating, it's making copies. It's also making the rest. Remember, the rest of the plasmid contains the bacterial DNA. And you have inserted your gene of interest. So as it continues to be copied, even the bacterial DNA is being copied. In a bacterial cell, the fragment of DNA from the human genome or other organism that is being studied is referred to as the foreign DNA. To differentiate it from the DNA of the bacterium, that is the DNA. What's the problem, Yusuf?
Okay.
Yusuf, can you mute your microphone? Okay, yeah. So we were saying, remember, as you continue to get copies of your DNA of interest, you'll also continue to get the rest of the bacterial DNA. And we are saying the copy of interest, maybe from the human genome or from any other organism that you're studying, that is called the foreign DNA. Okay. And of course, the one of the bacteria or the host is called the host DNA. You can grow up lots of plasmid carrying bacteria and use them as factories to make the protein that you're interested in. How will the protein be made? It will be made from the gene code of interest, because remember, it's from the genes that these proteins are made. And you can harvest the protein from the bacteria and purify it. Okay. Now, I want to stop sharing. So I want you to go into your groups that are set up very soon. And I want you to discuss, just from the little knowledge that you have already gained from those first few minutes, I want you to discuss why recombinant DNA technology is important in human development or in development in general. But I also want, you've already said the basics, what you're doing, we are playing around with genes. What are the potential dangers? It is good. I'm sure you'll tell me so many things that you think are so important and why maybe you are even interested in doing this. That is okay. That is perfect for me. But I also want to tell you to tell me what are the dangers when you start playing around with genes. There are some dangers. There are some things that people should be clear about. So we have 6 breakout rooms.
Madam, I have a question, kindly.
Ask.
Okay, as for me,
Or
actually,
maybe
I'm
what
not
you do,
Yes.
because what I want to do, as you do this activity, I want to see whether I can load the videos. Maybe we wait until you watch the videos and you do the discussion, and then now we ask the question, okay.
Yes, because I'm not understanding the importance of this recombinant DNA with human.
Okay.
And I don't, yeah.
That is why this group discussion is important. I just want to weigh how much you're understanding. Okay. So probably if you don't understand, your classmate has understood. Remember what I said, we are going to teach each other. So listen to your classmates as well. Ask, they will explain to you what they understand. We'll also watch the video. And then from there now, will you have understood? It's good to get this knowledge gradually, because once you get it gradually, it sticks. And then from there, if it is still not clear, you can still ask. Okay, so I'm opening the rooms right away. It is 15 minutes. So let's get to work.
Guys, una discuss nini man? Oe,
Watu ni aje? Waseya, hizi vitu mnaradanisha.
Ati nini? Ati unasema nini?
Hizi vitu mnaradanisha msee.
We na we.
Ama Google ina work.
I think people.
Huh?
So si zile vitu zenye tulikuwa tumepanga hata ni last time by the way.
So recombination basi hizo advantages ama sijui ni nini?
advantage ni kama inasaidia, unaona, kwa hii agriculture, development of drought resistant plants.
Tunaongelelea nini? The role of recombinant DNA.
Tunaongelea importance of gene recombination DNA na challenges.
Human welfare.
DNA to human development.
So si question ilikuwa inasomaje?
Recombination. The importance of Jaishika gene recombinant, like recombinant DNA technology in development, human development.
Recombinant DNA technology.
DNA technology in human development.
Importance ndio.
How it contributes to that one.
On this me there I think nilikuwa misunderstand your question. Me I was saying it from the other perspective of food production. So it's an aspect.
Si bado hiyo food production bado ni human development.
Food production, kuna hiyo food production na kuna development of vaccine, medicine. Lakini hizo ni zenye niko nazo, si I will google. Kuna development of vaccines.
Yeah, hiyo vaccine hata naiona hapa hivi, and pharmaceuticals.
Yeah, hizo ni zenye zina come to my mind. So the other bado iko na challenges. Challenges tunaanza na kwa food production. Food production inakata iko kwa in that line. Ukianza na food production, kuna issue ya insecticide venye sahi wana produce plant zenye ziko na insecticide. So inafanya agriculture iwe more reliant to, na kuna mic ya mtu inapiga kelele, inafanya agriculture iwe more reliant to pesticides since the pests are becoming more resistant to the earlier used pesticides. Then kuna another worry that interbreeding the two might become a challenge when the genetically modified plants interbreed with the native species. They come up with a totally different species which becomes a little more invasive. Those the species which are monitored. So hizo ni zenye ziko kwa akili saa tu we can search tuone.
Development of vaccines, growth hormones, antibodies, vectors, recombinant proteins, anti-cancer drugs. Hizo ni among the therapeutic products. Energy application inatumika kwa biohydrogen, bioethanol.
Buda, tupeleke polepole baba. Unaweza rudia tafadhali?
Gani?
Sasa nimesikia tu the growth hormones hapo kuna replacement.
See, sir, in different places, sir, medically, it helps in the production of vaccines, alafu in the production of growth hormones, hizo zinakaa zile manufactured growth hormones, alafu kuna antibodies, kuna recombinant protein na anti-cancer drugs.
Anti-cancer drugs. Nyingine
Kwa energy inakuwa useful in the production of biohydrogen, bioethanol, biomethanol na biobutanol. Alafu kwa agriculture it contributes to genetically modified vegetables, fruits. Ni kama zile maembe huanga hazina seed ama oranges zenye hazinanga seed. The rest kuna genetically modified microbes, genetically modified animals.
na sasa umemaliza by the way? Umemaliza?
Ndio sasa nimefika kwenyewe sasa. Kwa hizo my explanations mingi. A number of microbial strains have been developed which produce enzymes through specific engineering for production of proteases.
Hizo ni nini?
Hiyo ni application of recombinant DNA technology kwa food and agriculture. Helps in the production of proteases ama lysozymes, vitu kaa hizo maze. It is a wide nini, like that paragraph is so long. Kuna health and disease, kuna gene therapy.
Share screen bana.
Huh? Development of vaccines wenye tumesema, investigation of the drug metabolism, production of antibodies and their derivatives, I think we got enough points ready.
Tujasema potential dangers.
Yeah, sure, dangers.
See, I think dangers ni kama hizi yenye wamekuwa create na cancer. So hiyo haijakuwa proven. See, that might be the danger. Since the field is not that watu wajaingia vizuri and people don't know about the field that well. Kuna hii issue ya cancer causative cells zina come as a result of genetic modification.
Yeah, sure.
Then pia naona ni kama ina contributing contribute to kama ningesema I don't have a good name for it, but ina cause some invasive species. Seme mnafanya research then kitu iende mbaya and you end up producing something that is more dangerous than what we are trying to protect.
The production of alien products.
So that might be
Yes.
another cost. Then over time, due to use of pesticides which are genetically modified, kumekuwa na issue ya hizi I don't have a good way of putting it. So wacha kwanza
Tutaipush tu huko. Si Peter utapresent mse.
hapana.
Eh.
Mimi nitasoma hizo zenye mko nazo tu. Sawa, hakuna shida, nitapresent. Sawa, leteni point za dangers.
Okay, let's wait for everyone to return. And group 1, get ready. Let's wait for the rest to return. Is everyone back? Is everybody back? Okay, group 1. Group 1, can you tell us what you found out before we watch the video? Group 1 wako wapi?
We found out that recombinant DNA is widely used in biotechnology, medicine, and research.
No, it is, you guys stop being lazy. How in medicine? If you don't know, it's better you tell me you don't know, you've never heard, then you shall learn. But just telling me those two things, I think I'm not satisfied. Yes. All those 15 minutes, that's what you are discussing.
No, we are not done.
Oh,
so in medicine, yes, application includes it is used to detect the presence of HIV in a person. Like when someone goes for a HIV test, it is also used in the production of insulin.
Carry on, I'm listening like the rest. Yusuf, umemaliza?
No.
Because
it is also used in gene therapy in an attempt to correct gene defects which give rise to hereditary diseases. So people use recombinant DNA. It is also used in clinical disease classical example is where the application of recombinant DNA is possible.
Okay, so can you tell us some of the dangers? Just from this basic knowledge, I'm not expecting you to know much, but can you already see some dangers? Maybe just mention one or two things and then we go to the next groups.
As when recombinant DNA is used in agriculture, it will lead to production of super weeds, like weeds that resist to chemicals, and that could have an effect on agriculture.
Okay, thank you, Group 1. Let's hear from Group 2. Remember, I'm not expecting you to know much. We are just beginning. But I just want you to see, if you understood the concept, what are you thinking? How do you think it could be used? What do you think are the dangers? Group 2?
My name is Hassan Mohammed and I'm representing Group 2. On the importances of the recombinant genes, first we found out that it can be used in the production of vaccines and protein therapies such as human insulin. Second, we found out that it can be used in developing human organs, and it can also be used in the improvement of the genetic structure of human beings. And the possible drawbacks, first, as in it can lead to destruction of native genes, and it can also lead to vulnerability of disease, as in it can lead to vulnerability to diseases in human beings. Thank you, madam.
Okay, thank you, Group. The point you talked about growing organs and growing tissues. Is that really recombinant DNA? Because that I think is tissue culture, and in tissue culture, it's not actually recombinant. You're growing tissue from the original cell. So remember in lesson 1, when I was telling you there is the non-gene biotechnology. Sorry, I was muted. I'm saying, Group 2, that second point you talked about growing organs. And of course, skin is an organ. There has been a lot of, some of the breakthroughs in biotechnology has been to grow the skin tissue. But I was just raising a concern that let's be careful that we don't confuse gene biotechnology and non-gene biotechnology. When you grow organs, actually, that is not, you're not recombining genes unless you're bringing in a gene from another organism. But it's just, if it's just from the same organism, that is tissue culture and it's non-gene biotechnology. So let's keep that very clear as we continue, but thank you. Let's go to Group 3.
So we look at some of the advantages of recombinant technology, DNA technology, and the point revolves around the development of products of great benefits to humans. That is hormones, insulin, antibiotics, and vaccines. And also, in addition,
just a minute, Muya Mtuku, can you please mute? Muya Mtuku, please mute your microphone. Thank you. So Kamau, you can carry on.
And also, in addition, as far as health is concerned, it has been used to explore more on diseases such as cancer by looking at the gene in the genome which triggers the uncontrolled growth of cancerous cells. Additionally, to in terms of fertility on women, some hormones have been developed to trigger fertility. On concern, there are many. I would say time was less to discuss this, but the general concerns is we don't know the risk it poses. We know we are trying. It's an adventure. We are exploring, so we are not sure of the potential effects it has to the environment and to people in general. Thank you.
Okay. Sorry, have you finished, Kamau? Finish up.
Yes, I have.
Okay. Thank you, Group 3. Yeah. As we think about the importances, the benefits, in the hindsight, you always have to think about what are the dangers. Remember what I told you again, we are playing with the very, very essence of life, genes. And with all these combinations, what are you producing? At times, you're not even sure where this is going. So let's hear from Group 4. What did you think? What did you discuss?
Okay. I'm a member of Group 4. And we almost highlighted the same point as the other groups.
Please speak up, Clifford.
I'm saying we have highlighted the same point as the other group, Group 1, 2, and 3, particularly on biomedical application. That's the production of vaccines for cancer treatment, for diabetes, and maybe several plant disorders. And in addition, for another point, it can enable the creation of multiple copies of genes, that is by insertion of genes into other organisms to give that desired traits such as antibiotic resistance. And it has also improved food production in a way that it has reduced pest resistance, resistant to diseases, giving more production. And on the potential dangers, we have highlighted certain points. Destruction of the native species of the environment. That is by introducing the genetically modified species. They pose more danger to the native species through competition and other means. The effectiveness of these pharmaceutical compounds produced using the technology also has negative impacts to human health. They can cause inflammation, some ill health conditions, and certain disorders. And gene pollution of the environment has resulted to superweight antibiotic resistance. We also have some health effects from the food that is produced using the technology. And finally, the cross-contamination and the migration of the proprietary DNA between the organisms is also a negative impact.
Okay, thank you, Clifford, for that. So we're going to Group 5.
Thank you, Madam, for the opportunity. So in Group 5, we found out that recombination gene helps in phytoremediation and plant resistance development. Like when they are developed, of course, there will be developments of those plants that are resistant to drought and all those kinds. And we also found out that it helps in anti-cancer therapy. In anti-cancer therapy, like the development of those aids that help the cancer patients to battle it out and to live a healthy life. And in addition to that, we also found it useful in the development of vaccines and antibodies, which help in boosting human immunity system. And on the drawbacks, we found out that it helps, like it is disadvantageous to human health because some of these genes that are recombined can be harmful to human health and cause things like malnutrition in some cases, and in addition to some health problems, for example, cancers, because of the process involved. And also in biodiversity, it will be causing a competition, kind of a competition setup between the native species and the gene, the new introduced species, and also in extension, there will be interbreeding between the native genes and the introduced ones, which are genetically modified, which will lead to pollution of the gene pollution in the biodiversity. Thank you.
Okay, thank you, Michael Fares, for that. Peter Dua, you had raised your hand. Peter Dua?
Yes. So something just came to my mind.
Please speak up, we can't hear you.
So about cloning, the effect it might have to okay, the issue of cloning. Don't you think that when you have too much cloning, like for a species, let's say like a bean species, the plant might be susceptible to a certain disease, which might result in its extinction.
You're talking about cloning, Peter, and you're saying if you have too much cloning, what may happen?
That's what I'm talking about.
Cloning is a problem, and I think you're going to see it in the subsequent lecture. So let's just hold it right there. Once we continue knowing more and more about biotechnology and its applications, then we also we look at the ethics of biotechnology, what is allowed and what is not allowed. Okay, because there is what is allowed in biotechnology, there is what is not allowed. So at times you never know whether scientists are carrying out underground research. You see, most of the times when a government puts a hold or just say this kind of research is prohibited, those people who are keen and very curious might just decide to go underground. So you have underground labs that are just carrying out cloning maybe of human beings. I'm just thinking, I'm not saying they're research. Today morning I was reading about the theory, so far the theory of the origin of COVID-19. Of course, when the World Health Organization went to China, so this thing must have come from the wet market. No way it could have come from the lab. But they also said that they did not have access to a lot of the initial data. China refused to give them that data. And the other day, there was now a lot of speculation. People said, oh, we have received information that the Wuhan biosafety lab, four of the people who worked there had COVID-like symptoms somewhere in November, and they had actually to be admitted. Okay, of course, further strengthening the theory that thing may have escaped from the lab. What am I trying to say? There has been a lot of debate worldwide as to whether there is need to carry out such research. because what they do, these people, they just play around with those very highly contagious and lethal bacteria and viruses. Okay, they're really not playing around for the sake of playing around. They are doing research, they're saying, how can we deal with this virus? Should there be a pandemic? So what happens at times, there are accidents. There are accidents. Some of them leak out. Okay. So there has been a question, do we allow this to go on, or should all the countries in the world say no? And said, okay, if you say no, what you just do, you'll push these labs underground. And they still carry out the research, or some countries will secretly carry out this research without telling the others. So the same thing with cloning. It's almost, I think in all the countries in the world, it's not allowed, it's prohibited. But could there be some people who are cloning, maybe cloning the human genome? We don't know. We don't know. So we will come to a whole topic on the ethics of biotechnology, because as I say, it all looks nice, very many solutions, but remember you're dealing with the very, very essence, the last point in dealing with life. Taking an animal and putting part of an animal in a plant. There are people, of course, who are asking questions. So have that in mind as we invite group 6. Was there a group 6?
There is none.
Okay, great. If there was no group 6, let me try and share with you a video. I'll take it step by step because it will help make it more clearer what we have learned today, because this is the basis of biotechnology. Once you get it right, then you'll be able to make your argument. So I don't mind spending more time here and just making sure everybody is clear. So, and after this, then I'm going to ask still once again whether there is anybody who is still lost. So I'm sharing. Can you see the video? Are we together?
Yes.
Okay. So Leslie, I'll keep pausing and explaining.
Detail. Recombinant DNA technology is a technique that is used to refer identification, isolation, and insertion of gene of interest into a vector such as plasmid or bacteriophage to form a recombinant DNA molecule.
So I think that is what we have already learned. What is the definitions and the techniques and all that. And at the end of the day, you want just to produce as many molecules of the DNA as possible, or that gene, the fragment of interest, with the gene that is encoded by that.
And production of large quantities of that gene fragment or product encoded by that gene will be going through different steps in detail in this presentation. First of all, let us have a look into the synopsis of the steps in recombinant DNA technology. Step 1. It is the identification and isolation of gene of interest or DNA fragment to be cloned. Second, we have to introduce this isolated gene into a suitable vector. Then we need to introduce this vector into a suitable host. The process is called as transformation. We need to select the transformed host cell, and finally our indentation, multiplication or expression of the introduced gene inside the host.
So again,
let's move into the first step, that is identification and isolation of gene of interest. The introduced gene inside the host.
So again, the steps we have gone through, they're just being repeated So if you missed something, here it is in the video.
So let us move into the first step, that is identification and isolation of gene of interest. In recombinant DNA technology, from where we get this gene of interest, there are different resources for getting a gene of interest. These include genomic library, cDNA library, chemical synthesis of a gene. If we know the sequence, if the number of copies of a particular gene is very less, not sufficient for gene cloning, we can amplify that using polymerase.
Okay, remember me telling you that we actually now have DNA libraries. Yeah, they're already there. We've got the genomic library. You can get your gene of interest because some have been isolated already. We've got the cDNA library. I'm not sure what the C stands for. Then we've got the chemical synthesis of gene. If we know the sequence, you can, if you know the sequence, you can actually remember the what do you call it? The chemical, whatever, the guanine, the thymine, the four chemical, I don't know why that word is, whatever, of the gene, the four of them, I think the C, T, A, and G, if I'm not wrong. If you know the gene, for example, the code for the growth hormone, maybe it's a CTTA, then you can be able also to synthesize the gene chemically. You can actually go ahead and make your gene. And also, supposing you go to the library, to the cDNA library, and you don't get enough copies of the desired gene, then that's when you go for cloning, as we have explained before. And then you go for gene amplification like PCR. We are going to do that later.
Chain reaction, gene amplification technique. So in order to understand all these concepts, let us take an example. Here we are going to introduce human insulin gene in bacteria. Step 1. From genomic library, we have taken human insulin gene. As I mentioned, genomic library is a source from where we get our gene of interest. So step 1 is I have taken human insulin gene from genomic library. Step 2, I need to insert this desired gene, that is the insulin gene, into a suitable vector. Before that, we need to know something about vector. What is a vector? Vector is any DNA molecule that has the ability to replicate inside the host to which our gene of interest has integrated for cloning. Vectors include plasmids, bacteriophages, bacteria, etc. You can refer our video gene cloning vectors for more. So, most often, here we are taking plasmid as a vector. Plasmid is extra chromosomal DNA that is present in bacteria. This is E. coli bacterium. I have isolated plasmid from E. coli bacterium. This is a plasmid vector. So this is my gene of interest. This is our gene of interest, that is human insulin gene. So first of all, we need to make a cut in the vector using a restriction enzyme. Restriction enzymes that are capable of making internal cuts at specific sites in the DNA molecule. So we need to incorporate our insulin gene into this vector. So what we did was, I made a cut using restriction enzyme, and now I have the gene. So I have incorporated my insulin gene into the vector, and I sealed the gene, the cutting ends with the enzyme ligase. Now we have the recombinant DNA molecule or primary DNA. A recombinant DNA molecule is a DNA molecule from which DNA from different sources. You can see right here we have a recombinant DNA molecule with human DNA and bacterial plasmid DNA mixed together. So the recombinant DNA molecule is just a recombined DNA molecule from different organisms. So second step is over. We have inserted our human insulin gene into a vector. Now the third step, introduction of this vector into a suitable host. There are different gene transfer methods by which we can introduce our vector into a host. These are some of the methods: electroporation, microinjection, liposomal-mediated gene transfer, silicon carbide fiber method, etc. And hopefully we'll be discussing all this later. And Chemical gene transfer methods are there. PEG-mediated gene transfer, calcium chloride-mediated gene transfer, extran-mediated gene transfer.
In this particular part, we didn't look at this, the introduction of vector into a suitable host. And why are you introducing a vector into a host? For the purpose of replication. This is where the cloning takes place. And as you can see, there are various methods. There is a physical transfer method where you can use